When you think about the human brain, probably an image comes into your mind that looks something like this. It's a continuous sheet of folded cortex comprising many, no one really knows how many neurons and, of course, many other types of cells. However, for the remainder of your career as an EEG or MEG researcher, you should think about the brain not as a continuous sheet comprising neurons, but instead as a surface that contains many independent nodes or vertices, each one functionally homogeneous. So let's see what this looks like. We can zoom in a little bit here into the brain. And now we want to be thinking about the brain as comprising functionally homogeneous, you might call these columns or hypercolumns, or we can just refer to them as tubes. So these are like tubes in the brain. Each one is approximately 1 to 2 millimeters in diameter. Each one contains somewhere around 10,000 to 100,000 neurons. And all of the neural dynamics and neurophysiological and biophysical dynamics that are happening inside each of these tubes is the same. It is functionally homogeneous, what's happening inside this tube. Of course, this is not entirely realistic. In the real brain, there are all sorts of dynamics that are happening at a spatial scale smaller than this. However, this is approximately the smallest spatial scale that can be measured with EEG or MEG. So this is how you should try to conceptualize the brain when you are interpreting your EEG or MEG data. Of course, if you're measuring LFPs, or you have electrodes that are implanted inside the brain, the spatial scale can be considerably smaller than this, but this is about the smallest spatial scale that you can resolve with EEG or MEG. Okay, and each one of these tubes is producing electrical field or magnetic fields that you measure from outside the head. Now, typically in your experiments, you record not only from one electrode, but from many electrodes. This is a visual representation of the locations of electrodes from a 64 electrode cap. And hopefully going into the future, 64 electrodes will be seen as kind of the minimum number of electrodes, and something like 128 or more than 200 electrodes will become increasingly widely used and become standard. Nonetheless, so here you see 64 electrodes laid out on a two-dimensional representation of the head. This is called a topographical plot or a topographical map. And in case you are not familiar with interpreting these plots, what you want to do is imagine you are looking down on the top of someone's head. And here is the left ear and the right ear, and this is the nose. And now these are the locations of the electrodes, and now what we want to do is try and understand how, or to visualize how the activity is changing over the entire scalp, over the entire surface. So of course the data that you record from any one electrode might look something like this, and most of the tutorials and lectures from this point on will really deal with how to analyze and how to extract information out of the time series that you get from a single electrode. But in practice, you really want to look at the change of activity over space as a function of space. So there are a few different ways of representing the spatial information that you get from EEG data. Here you see probably the most conservative visual representation. So the activity of each electrode is mapped onto a color, or in this case, a gray intensity value. So here you see, let's imagine that black is mapped onto lower levels of activity and white is mapped onto higher levels of activity. This is a very conservative way of illustrating the spatial distribution of the data, because we are not making any assumptions about what the activity might look like between these electrodes. But on the other hand, it's also a little bit difficult to interpret this plot. So by far the most typical way to analyze or to illustrate the spatial distribution of EEG data is with these interpolated two-dimensional topographical maps. And in a few minutes, we'll switch to MATLAB and I will show you how to make these topographical maps. But the idea is that we don't actually record any data from this point here. This is a point that's in between these electrodes where we actually record data. But we want to use an interpolation algorithm to estimate what the activity would look like at this point, and this point, and this point, and this point, and so forth, in order to produce a smoother plot, which is easier to interpret. Here you see this same activity, so actually all three of these plots show exactly the same data, just represented in different ways. Here you see the representation on a 3D rendering of a head. So there's some advantages and disadvantages of three-dimensional topographical plotting versus two-dimensional topographical plotting. Of course, the three-dimensional plots look much nicer, they make nicer figures, and they're certainly more directly interpretable in terms of the anatomical distribution of the activity. So it's much easier to see where on the head these different features are coming from. On the other hand, of course, you only see half the head, or even a bit less than half the head here. So if you wanted to show what the entire spatial distribution of activity looked like, you would need at least two different 3D plots, and maybe even three if you wanted to show the activity here all the way posterior and here all the way anterior, but you would certainly need more than one to show the entire head. Two-dimensional plots, on the other hand, have the advantage of showing pretty much the entire spatial distribution for the entire head on one plot. Sometimes they get cut off. This is a parameter for how much of these data coming from behind the head you want to show here. But they show pretty much the entire topographical distribution, but they're a little bit more difficult to interpret in terms of the anatomical scalp localization of the effects. Topographical maps are very important. They are very powerful and should be used very often. In particular, not only when you are inspecting your results and showing images of your results after you've completed your analyses, but also during pre-processing and during the cleaning and initial inspection of the data. Topographical maps are very powerful, and you can extract a lot of meaningful information very quickly. And so here we see two topographical maps. This one, all I'm telling you is that it's a topographical map of the activity at 130 milliseconds, and I'm not telling you anything else about this. I'm not telling you what happened at time zero, but you can already come up with a pretty good guess just based on looking at this plot that at time zero there was a visual stimulus that was presented. And you might also guess that the stimulus was presented on the right side of the screen, so in the right visual hemifield. And in fact, that is what happened in this experiment. There was, at time zero, a stimulus presented on the right side of the screen. This is very useful. For example, if this was the first subject in your pilot experiment that you were testing, and let's say this is supposed to be the condition where there was a stimulus presented on the left side of the screen. So now, this should raise some alarm bells that perhaps something went wrong with the experiment trigger coding, the labeling of the markers or something. So it provides a bit of a sanity check looking at these topographical maps. Here you see at 400 milliseconds. Now, again, I'm not telling you what happens at 400 milliseconds, but you can probably guess by looking at the plot that it's likely that there was a button press. Maybe there was even a right-hand button press. Maybe the subject pressed the button with the right hand at somewhere around 400 milliseconds, and that's reflected in his motor activity. And in fact, this is what happened in this experiment. At somewhere around 400 milliseconds, there was a response using the right hand. So already these topographical maps are telling you a lot of very important and detailed information that you can use during pre-processing to check and make sure that the data are good and clean and consistent with what you should expect based on the design of your experiment. Topographical maps are also very useful for identifying potentially bad or artifact-ridden electrodes. Here you see a series of topographical maps. The numbers here correspond to time in milliseconds, so this is a quarter second after stimulus onset. And it's very clear, it's very obvious that something is wrong with this electrode. You can see it has very large values and they're very different from the neighboring electrode. So we know immediately by looking at these topographical maps that something was wrong with this electrode. So we need to resolve this issue before going any further with these with the data analyses. I would like to clarify two terms that are kind of related and a little bit easy to confuse, but they are actually, there is an important difference between them. And the terms are topographical localization versus brane localization, also sometimes called source localization. The difference between these two terms is illustrated by these two pictures here. I apologize for the really ugly pixelated nature of this topographical map, but it was very small in the figure and I had to blow it up. Anyway, topographical localization is really just an observation of where your effect is maximal on the scalp. So here we see the effect, whatever this is, this statistical map, this condition contrast, is larger at this electrode than it is at any other electrode on the scalp. So we can localize the effect to this particular electrode as opposed to any other electrode. With topographical localization, we are not making any assumptions. We don't need any special analyses. We don't need any specific sets of parameters. We are really just observing and commenting on where an effect is empirically observed to be localized to on the scalp. That's a different question. We don't necessarily know where in the brain this dynamic came from, where in the brain the source is. All we know is that it is maximal at this particular electrode. Brain localization, of course, we want to make an inference about where in the brain this activity may have come from. And brain localization or source localization does require specific algorithms like dipole fitting or beamforming or any other source localization algorithm. Source localization requires some assumptions. It requires several parameters. So, and the inference is different. Here we are trying to make an inference about where in the brain did the activity come from that produced the observed scalp topography. And here with topographical localization, we are really just asking the very simple and definitely answerable question of which electrode shows the effect the biggest. Okay, so now I'd like to switch to MATLAB here. If you download the sample EEG data and the code that is presented along with this video, you can follow along with this script. So what we're going to do here is, or what I'm going to do is introduce you to topographical plotting. Basically, I'm going to, let's see, show you how these kinds of topographical plots are produced in MATLAB. Let's go MATLAB. The first step that we're going to do is transform the Cartesian coordinates to polar coordinates, which is important for the next steps. Once we have that, we can plot the XY locations of the electrodes. And here I'm using a function called scatter, which is very similar to the plot function. In fact, you could produce exactly the same plot with the plot function. It's just sort of shown here just for your personal edification. So this should look familiar. This is just the layout of all the electrodes. This is very similar to what I showed in the slides, except that in the slides that was produced from an EEG lab toolbox function that basically just plots these electrodes plus adds some circles for the head and the ears and the nose and so on. But this is just the electrode locations. What we are going to do next is define points on this XY plane where we want to interpolate the data. And so I encourage you to play around with this and change the interpolation detail. Here I'm just using arbitrarily, I just arbitrarily picked a value of 100 as the level of detail. So we are going to interpolate 100 points between the minimum and the maximum of these xy plane coordinates. Then this function mesh grid just converts them from a one-dimensional array to a two-dimensional array, which is important for the topographical plotting. So here, all the black dots, this is where we're going to estimate activity. So the red dots are where we actually measured data. These black dots, we don't actually have data. So in some sense, or in a very strict sense, we don't actually know what was happening on the scalp at this location. However, we are going to use a weighted combination of all these other electrodes to estimate what the activity would look like at this point here. And that is going to allow us to create a topographical map with more detail than just these individual 64 data points. So here you can see I used another plot. This is also, it could have been done using the normal plot function, but this is just to introduce you to some other of MATLAB's functions. So plot 3, we plot the x and y coordinates, and here I'm plotting the z coordinates as -1, which just plots the black dots behind the red circles. So if you just use a plot 2D function, then you would just see that the black dots would be on top of the red circles, which I just think looks a little bit less nice. So you can see it would look like this. It's basically the same. Okay. Now what we are going to do is compute the average activity at an arbitrary time point. So I picked 100 milliseconds here. And we learned in a previous video that this is the function or one method that will allow us to convert between the time point in milliseconds that we want and the time index. So here we know that the 283rd time point in the EEG data corresponds to 100 milliseconds. Okay, and then we want to extract the average data from this time point. So we take EEG.data from all channels from this time point, from all trials, and then we take the mean over the third dimension, so that gives us the trial average. This particular interpolation function requires that the input data be double precision, or floating point precision, and so the function double here is just to make sure that whatever comes out of here will not produce an error during interpolation. Okay, and so this tri-scattered interp, this is a MATLAB function that it provides us with a function to compute interpolation. So now we're going to interpolate over these grid points, so all these black dots here, to get a 100 by 100 map of interpolated points. And the 100 by 100 comes from this parameter here. So you can change this, you can try setting it to 1,000 or 10 and see what the difference is. Okay, so now that we have our interpolated data, we are going to plot this interpolated data, and I'm just plotting it in a few different ways just for comparison. Here I'm plotting it using the MATLAB function contour f. The f stands for filled. Here using surf, which stands for surface. And you can see that these appear to look identical. The only difference is with a surface plot, we are actually representing this piece of data as a surface, as a landscape. So you can see that the Z values, the microvolt values, are mapped here onto color. What I like about this representation is that it helps you understand that these features over multiple dimensions, these multidimensional feature spaces, can be conceptualized as landscapes, as a terrain, so to speak. The contoured function actually is just a flat function. So from when you're looking down, of course, it's the same as a surface. image using the function image or image SC, which means it just does some color scaling. You can see two differences between image and contour. The first is that the image function appears to be a bit more pixelated, whereas the contour appears to be a bit smoother. And the second and more important difference is that the direction of the y-axis is flipped. So here this red feature is on the bottom, and here it's on the top. You can see when you look at the y-axis that it really is the same data. This is just flipped. Image by default shows in ij coordinates instead of xy coordinates. So it just has the smaller numbers at the top. It doesn't really matter whether you use image or contour, but it is important always to remember that image is going to flip the y-axis relative to contour. This is very important when plotting time-frequency plots because you want to make sure that you are labeling the y-axis correctly, right? You don't want to have the lower frequencies on the bottom, but you mistakenly think that the lower frequencies are on top, or vice versa. It's easy to get confused, so you just always have to make sure that you're careful with the image SC. In this figure, you see the interpolated data using the EEGLab toolbox function called topoplot. So you can see these, it shows the same data, but of course they've added all these other nice features like the circle for the head and the locations of the ears and the nose and things. Another difference you can see is that EEGLab topoplot function is using a different interpolation function. They use one called grid data. Here I used one called triscattered interp. The difference is that this function triscattered interp only does interpolation. It doesn't do extrapolation. And what that means is that here is the boundary where we recorded our data, and anything within this boundary would be interpolation. Anything outside this boundary is actually, it's not possible to interpolate because there's nothing to interpolate to. So instead, estimating a point out here would be extrapolation. And so you can see that these values are all NaN, so they just don't exist because these would be extrapolated points. With this grid data function, it's doing both interpolation and extrapolation. So it's also just another difference to keep in mind. So I hope that helps you understand and be able to interpret where topographical maps come from and how data can be represented from multiple channels over space.